Bismillahirrahmanirrahim. Assalamu alaikum. Chapter number 16, Inherited Chain. The lecture number 4. This is the interaction between the locus. Genes on the same chromosome are linked to each other, and this is called the gene linkage. And the number of linkage groups is equal to the number of the homologous pairs of the chromosomes. For example, human has 23 linkage groups. Genes of color blindness, hemophilia, GOT, etc. These are these form the linkage group on the X chromosome. The genes like the sickle cell anemia, leukemia, albinism, these are found on the 11 chromosome, that is the autosome. So the linked genes whose loci are close to each other do not obey the Mendel's law, which we have done yesterday, of the independent assortment. So because they cannot assort independently during the process of meiosis, because they are too close to each other. That's why we can't separate them during the process of meiosis. So the gene linkage minimize the chances of genetic recombination and variation. But there is a process of crossing over in which the linked genes can be separated by crossing over. And closer the two loci, more strongly are their genes linked, and further apart two genes lie, greater their chance of separation through the crossing over. So crossing over is the exchange of segments between the two non-sister chromatids of the homologous chromosome during the process of meiosis, and we have done this in the prophase uh, 1 of the meiosis 1. Now this is the this is the diagram of showing the crossing over, or which recombines the genes. So these are the meiosis chromosome, and these are the meiotic products. Actually, this is a chromosome, and this is a chromosome. This is one homologous chromosome. This is one homologous chromosome. This one homologous chromosome has genes AB and AB, capital AB, capital AB. This one has the small ab, small ab. Meiosis with no crossing over between the genes. So what will happen when these are separated? They were sharing the same centromere at that position, but after the process of meiosis, they get separated. What will happen? This is actually showing the parental type. This is showing the parental type. This is showing the parental type. This is showing the parental type. The chromatids are separated at the point of the centromere. This is the centromere. And after the process of separation, these products all have the parental types because they have not undergone the process of crossing over. But another example in which there is a process of crossing over, for example, this is one chromatid, this is the second chromatid. What will have, this is one chromatid, this is the second chromatid of the homologous chromosome. This is one homologous chromosome, this is one second homologous chromosome. In the process of crossing over during the meiosis one, in the prophase one, they have exchanged the segments between them. And by exchanging the segment, what will happen? When they get separated at the point of the centromere, after the process of meiosis, what will happen? This chromatid is of parental type because there was no chiasmata formation over there. This will also be of parental type because there was no chiasmata formation, pairing of chromosome. But at this junction, at this bridge formation, this chiasmata formation, there will be the exchange of segments. They have exchanged their segments likewise, capital A, small b, instead of capital A, capital B, and small a, capital B, instead of small a, small b. So these two chromatids are of recombinant type, or they have experienced the process of crossing over. Recombinant type, they are the new ones, they are different from their parent strand, these strands. This is the process of crossing over, which will recombine the genes. Now we can judge the recombinant frequency or crossover frequency with the help of this example, that is directly proportional to the distance between the linked gene locus. How this is formed? We can take the recombinant types divided by the sum of all the combination into 100. How? There's an example which will clear all these things. For example, there is a chromatid capital A, capital B, a, small b. These are all the strands these are strands. This is parent 1, parent 2, parent 1, parent 2. They have been crossed, and these are the gametes which are formed after the process of the, you can say, crossing over, because they have done the exchange of segments like this, in which we have done in the previous example, like this. These are the recombinant types, these are the parental types. And we are going to cross because these two are same to each other. A small b, small ab, these two strands are similar, and these two strands have experienced the process of crossing over. So we are going to cross this strand with all these gametes. How? We have taken this one that is similar. Because that is similar, we are going to take only one chromatid, small ab, on this side. And that is the capital AB, this one, small ab, this one, capital A, small b, this one, and small a, capital B, this one. And we are going to make a cross between all these. So small a, b with this one, resultant, this one with this one, results, this one with this one, and this one with this one. These are showing the results of the crosses. What will happen? For example, in any example, for example, we have got the parental type, that is 40 plus 40. These are two parental types. Now, for example, we have got out of 100, we have got 40 this one and 40 this one. These are this one. Total is 80. And we have got the recombinant types, for example, this one and this one. These are, for example, 10 plus 10 is equal to 20. So the recombination frequency will be 20 recombinant type divided by the whole sum. This one, sum of all the combinations, 40 plus 40, 10 plus 10 into 100. The answer is 20. That is directly proportional to the distance between the linked gene locus. For example, we have this combination. It means that these two genes are present 20 units apart. 20 units from 0 to 20. 20 units are apart. This is showing the crossing over frequency. For example, if that value is 10, it means that the genes are very close to each other. 20, it means they are not close to each other. 30, they are not close to each other. For example, we can judge that how uh, close these genes are, or their locus are present on the same chromosome or on the same chromatid, right? So this is how can we judge the crossover or recombination frequency. Now we can have a chi-square test. Uh, that is actually used to compare our observed results with the expected one. For example, we have uh, done in our law of independent assortment, that is the ratio is 9 ratio 3 ratio 3 ratio 1. We have the probability of 9 divided by 16, 3 divided by 16, 3 out of 16 and 1 out of 16. And if there is the significance difference between the two, for example, our results are not matching with the expected one, and our practical results are not matching with the expected one. So how can we detect the difference between the observed values and the expected values? We can use the chi-square test. That is sum of the difference between the observed and expected square divided by the expected results. So we are going to do that is, for example, we have got any kind of, you can say the difference between any kind of two results. For example, we are taking the lengths of corolla of any rose species. Then we have got the chi-square test 0.79. The answer is, for example, that is 0.79. Suppose we have got the chi-square result is 0.7. For example, we have four values. Four values mean we have taken four different results or four different phenotypes. 9 ratio 3 ratio 3 ratio 1. We have four classes. So actually n is the number of classes, and the degree of freedom means over there n minus 1. So our degree of freedom in this case, in this example, we have 4 minus 1 is equal to 3. That is our degree of freedom. And we have been given a chart like this, in which there is a probability greater than this one. The chart will contain the values of 0.1 means that 10, 1 out of 10. It means that is showing more error between the observed and the expected values. 0.05 means there is a kind of error of 5 among 100. It is very negligible. Or 0.01 mean 1 out of 100 error, or 0.001 mean 1 out of 1000. That is very negligible amount. So in biology, we take the 0.05 probability. So what will happen? We will take the degree of freedom. For example, in this case, we have used the four classes. 4 minus 1 is equal to 3. We will take 3 over there. And we will take 0.05 as our standard. And we will match this column in 3 degree of freedom according to our example, match with the 0.05, and the value stands over there 7.82, and we have got the value 0.79. Suppose this 0.79 is very small than the 7.82 which has been given in 0.05 along the degree of freedom 3. So it means that the difference between our observed and expected results or value is not significant, and that is due to by chance. Actually, our value 0.79 doesn't lie at any place over there in this whole chart. So it means that value doesn't lie in any this you can say the chart or this table. So it means that the difference between the observed and expected values is non-significant and due to chance. So it means there is no difference, no significant difference between our observed and expected values which we have done. Observed mean which we have done with the help of our practical or observations of by collecting the data, an expected one. So we can prove this with the help of chi-square test, and this test is in your chapter number 16, and you can see the example from there.